DNA is the instruction manual for how to build life. From animals to plants to humans, it defines us all. Information is stored in DNA using 4 types of molecules, which come in pairs. We have billions of these pairs. DNA folds itself into paired packages called chromosomes, which are stored in the nucleus of cells. Each species has a different number of chromosomes. Humans have 46 chromosomes, which contain our genes. Genes act as instructions for molecules called proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases, and we have an estimated 20,000 to 25,000 of these genes passed during reproduction. Each person has 2 copies of each gene, one inherited from each parent, and most genes are the same in all people except for a small number of genes being slightly different. These small differences contribute to each person's unique physical characteristics, varying from height, color, and personality traits. Some genes are dominant and some are recessive. Dominant genes are more often expressed as observable characteristics, while recessive genes are often masked by dominant genes. The recessive genes are expressed when inherited from both parents. And while all humans share over 99% of our DNA with each other, what we don't share is what makes us unique in our own way.
Hey guys, I hope that short video gives you an idea of where we're going next. We are moving away from cells and into DNA and genetics. This is always everybody's favorite unit. We're going to talk about how traits are inherited. But in order to understand genetics, we first have to understand the structure of DNA. And that can be kind of difficult because different diagrams show DNA in different ways. So, for example, I have a few up here for you. Here's one diagram of what DNA can look like. Okay, it can also be a little more simplified and look like this diagram here. And you can see the base pairs and the backbone. And the backbone has kind of been simplified here. It's just all one color. Or you can have a more folded out chemistry version of DNA. And my goal for you is by the time we finish our DNA structure lesson tomorrow, you'll be able to identify the structure of DNA no matter what type of diagram is given to you, and also that you'll be able to know the purpose of each part in DNA. So here's a little introductory video for you. We'll discuss more tomorrow in class. Thanks, guys.
Hey guys, I've put together a DNA model for you and I just wanted to go over the structure of DNA with you before you moved into your next. The first thing I want to show you is in your textbook. Okay guys, this is the figure that I wanted you to take a look at. It's page 280 in your book, figure 10.5 if you want to go back and take a look at it, and it shows you how eukaryotic chromosomes are organized. So if you look over here, okay, DNA is a double helix. Okay, it coils around these proteins called histone proteins, and the DNA and the histone together is called the nucleosome. And then you can see that that complex curls again and curls again into supercoils until we form this tightly coiled, or what we may call a condensed chromosome. And remember, the purpose of that condensed chromosome is to ensure that when the cell divides and when this chromosome is split in two during anaphase, that each new cell gets an equal copy of the DNA. So this chromosome, the purpose of this chromosome structure is to organize the genetic information, organize the DNA in a way that can be passed easily into the new cells. So what we're going to do today is we're going to zoom in and we're going to look more at this DNA structure and talk about the purpose of the different parts of the DNA molecule. Take a look at this handout, but I wanted to walk through this with you to talk about the different parts of the DNA molecule. And you can see that the molecule I'm showing you here doesn't look exactly like the one you just saw in the book. What you just saw in the book is often referred to as like a ladder molecule. It's kind of simplified. It shows you like the center of the ladder is the nitrogenous bases, and then the backbone is kind of melded together as the sides of the ladder. So what this diagram is doing for you is it's completely expanding out that DNA diagram, and you've got your different parts of a nucleotide, different parts of the DNA molecule, and you can also see the bonds. So this is just a different type of diagram that you might see. If you look, notice that you'll hear me refer to the rungs of the DNA molecule and the sides of the DNA molecule. So on the rungs of the ladder, we have alternating phosphates and sugars. And remember that in DNA, the sugar that you have is deoxyribose. It is a pentose sugar, meaning that it has 5 carbons. Okay, so the backbone are these sugar phosphate, these alternating sugar and phosphates. They are bound together by very strong bonds. Okay, strong covalent bonds, because they're protecting what's on the inside, which is the precious code. These nitrogenous bases are going to be the key. Okay, they're going to be the directions for a protein. And so they are protected within this sugar phosphate backbone. And I'm going to show you in a minute how the coiling and the double helix shape can help protect these molecules. If you look in the center, okay, you have 2 bases bound together. Okay, adenine, remember our stick letters go together, our stick letters bond together, and our curvy letters bond together. So adenine bonds with thymine, and cytosine bonds with guanine. Okay, and I've got another cytosine down here. And it won't always look like this. The bases that you have will depend on what the code is. The other thing I want you to notice here is that some of your bases are bigger than the others. Adenine and guanine are larger than thymine and cytosine. The difference in this is the size and the type of bases. So adenine and guanine are what we call purines. Okay, and what that means, you can't see it in this diagram, but both of these are ring structures, okay, and the adenine and thymine are double rings. So in my attempt to draw, it's going to look kind of like this, except for they're a little bit more the same size. So this is your adenine and guanine. Okay, so they are larger. Okay, whereas your cytosine and thymine are going to be single ring structures. Okay, these are your pyrimidines. Okay, and because they are a single ring, they are smaller. And for the longest time, I had a really hard time remembering this. So the way I try to remember this now is that the longer the name, the smaller the ring. Okay, so pyrimidines, the longer word is a single ring. Purines, the shorter word is longer rings. The other got purines and pyrimidines, and they bind together, okay, by hydrogen bonds. And if you'll notice, guanine and cytosine are bound by 3 hydrogen bonds, whereas adenine and thymine are bound by only 2. So this guanine-cytosine bond is actually stronger because there's more bonds there. However, overall, these are weak hydrogen bonds. And the purpose of that is they are easy to unzip or break those bonds. And these bonds are going to be broken at 2 different times during the cell. They're going to be broken during DNA replication, where all the DNA in the cell is copied. They will be broken then. This molecule will be split in 2, and each side will serve as a template strand. And they're also going to be broken during transcription. Okay, when transcription happens, when an RNA copy is made, this DNA molecule unzips down the center, and one strand becomes the coding strand, or the strand that will be transcribed into RNA. So these bonds have to be zipped and unzipped quite often because your cell always needs proteins. Transcription is always happening within the cell. Okay guys, just a couple more things that I want to show you. The subunit of a DNA molecule is a nucleotide. Okay, and as you can see, each rung in the ladder is 2 nucleotides. A nucleotide is a phosphate, sugar, and nitrogenous base. So here we have, let me switch pens. Okay, here we have 1 nucleotide. So if you look on the DNA molecule I'm showing you, I've got 1, 2, 3 rungs, and again, that's just an analogy. It's not a technical word in DNA, but I've got 3 rungs in my ladder, okay, which means I have 6 nucleotides because each side of the DNA has 1 nucleotide. Okay guys, so the last thing I want to show you today is this awesome DNA model that I put together so that I could show you something 3D. But this is a 3D molecule that I have put together, and right now I have it in the ladder form, meaning that I have the DNA untwisted. It's not coiled, it's not in a double helix right now. And from that you are able to see our bases. Okay, and notice that one base is larger than the other base in each pair. So these are my purines because they're larger, okay, and these are my pyrimidines because they are smaller. So this, for example, is guanine because it's bigger, and cytosine. And this is adenine and thymine. And use some of those little word tricks that I gave you to help remember the difference because it is really difficult to remember. So my bases are in the middle, and then if you look on the side, it's a little bit difficult because it's clear, but you can see the alternating sugars and phosphates. So my sugars here are white and my phosphates in this model are clear. So if you look, the way you can always find the sugar is that the sugar is going to be touching the base. Okay, so down the side, it's going to go phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. And each of those is a nucleotide. Okay, the other really cool thing about this model is that I can show you how it coils. I can show you the double helix, so let me do that. Okay, so this is me partially coiling it, and this is how you'll see it a lot of times. In diagrams, this partially coiled model, but I can also coil it all the way up. And when I do that, you can see, and again, it's a little difficult because the phosphates are clear, but when I do that, you can see that my genetic code, okay, my bases are completely protected within the phosphate sugar ladder. And that is the purpose of that strong backbone, is to protect this code. Because what you're about to learn next is that this code is precious, okay? It is the instructions for making all the proteins in our body. So it is protected by these sugars and phosphates. It is super coiled, okay? And my bases are protected here on the inside. Okay, and then when it's time for either DNA replication or transcription, let me see if I can do this. Whoop. Okay, the molecule unzips and unwinds, okay, or I should say unwinds and unzips. And of course I won't be able to do this in real time, but these break apart and it unzips the molecule. Okay, so ta-da! Okay, here's my molecule unzipped. These weak hydrogen bonds have been broken, and now the code is exposed. So if this was DNA replication, each side would serve as a template strand. New nucleotides would be added, and I would form 2 new DNA molecules. Or if this molecule has been unzipped for transcription, then the RNA will be formed from one side of this exposed. That was a review of DNA structure. Please let me know if you have any questions. Thanks, guys. Biology course, but the